Hello everyone, my name is David Matis and I will be talking about aseptic techniques and use of media. Aseptic technique and use of media. Appropriate aseptic technique and use of media is crucial during lab experiments to prevent microorganisms from contaminating samples and culture medium. It also allows for reliable and accurate results, allows for a focused study and analysis by isolating specific microbes. Here I will be talking about aseptic practices, such as disinfecting your work area before and after your lab experiment, the use of lab coats and gloves, flaming the top of sample tubes before and after collecting the sample, slightly opening the culture lid when transferring microbes, and use of new inoculation loops for different samples. And here to your right is a variety of images that will help keep aseptic techniques. Objectives of this lab were to apply aseptic practice during lab experiments, prevent contamination of harmful microorganisms to your samples, maintain the culture purity, study and observe the growth of isolated microbes during your lab. Alright, I'll be covering materials and methods. Some materials used in this lab were inoculating loops, liquid paraffin candle, grease pencil or Sharpie, culture tube rack, triptych soy agar plate, triptych soy broth tubes, triptych soy agar slant, sterile agar slant, and lyophilized Certia marcinis. Next, we'll be talking about methods. This will be talking about activity 1, aseptic reactivation of a lyophilized culture. Here are steps 1 through 13. Activity number 2 is aseptic transfer from broth culture to broth. Activity number 3 consists of aseptic transfer from broth culture to slant using aseptic technique. Activity number 4 is aseptic transfer from broth culture to plate. These are my results from activity number 1 from the lyophilized culture reactivated. It was incubated for 48 hours and it was noted to be a successful reactivation by the turbidity, which is cloudiness in the tube. These are my results from activity number 2. It is a transfer from broth culture to broth, and it was incubated for 48 hours. Just like activity number 1, the sample tube on the right has a turbid color. These are my results from activity number 3. Transfer from broth to culture slant, incubated for 48 hours. Bacterial growth is noted on the slant as the image on the right demonstrates red streaks. These are my results from activity number 4, transfer from broth to culture plate. It was also incubated for 48 hours. Bacterial growth was observed on the plate as the image on the right shows streaks of red microbes growing. Lastly, I will be discussing the importance of aseptic technique. The purpose of practicing aseptic techniques with different types of media is to learn how to prevent microorganisms from contaminating samples. It allows for reliable and accurate results. Aseptic techniques are not only crucial in microbiology but also in healthcare. Aseptic techniques help in preventing hospital-acquired infections from spreading. For example, in the medical field, it is standard of care to use sterile technique when inserting a urinary catheter in patients. By using sterile technique, the patient will have a decreased risk of infections such as UTI. It is also standard of care to wash your hands when you're coming into a patient's room and when you're leaving a patient's room. My results relate to a broader understanding of microbiology by performing and understanding various techniques that help prevent cultures from being contaminated with unwanted microorganisms that are not able to be seen by the naked eye. It also provides an understanding of how to study, manipulate, and isolate certain microorganisms for study.